Hi, my name is Annie Chu, and I work at the National Institutes of Biomedical Imaging and Bioengineering under the mentorship of Dr. Alexander Gorbach, and my project is titled Skin Temperature Oscillations in Patients with Sickle Cell Anemia. So first, I'm going to talk about the disease I studied. Sickle cell anemia is a hereditary blood flow disorder that can change the shape of your blood cells from their normal healthy circular shape to a crescent moon-like shape. And this is really bad for blood flow because it can obstruct it and cause blood not to be able to pass through the vessels. And it also leads to many painful symptoms such as pain crises and ulcers, and there's currently no cure for the disease, even though you can mediate the effects with different drugs. So particularly, we were interested in the oscillations of blood flow, which are controlled by many different mechanisms throughout the body, and each of these mechanisms are manifest themselves in a different frequency range. So first we have the myogenic frequency range, which is the oscillations of blood flow controlled by the smooth muscles, and these muscles cause the blood vessels to dilate or constrict, controlling how fast the blood is able to flow. Next is the sympathetic range, which is the sympathetic nervous system in your brain that sends signals to the vessels to whether to control the rate of blood flow. We have the endothelial frequency ranges, and these are controlled by the endothelial cells in the blood vessels, releasing different substances, telling the vessels to dilate or constrict. This is further separated into different categories based on whether the substances released are nitric oxide dependent or nitric oxide independent. So the primary goal of my project was to determine if there were any differences in the skin blood flow oscillations in sickle cell anemia patients compared to regular healthy people, and also to see the effects of occlusion of blood flow on the arm and how this affects the blood flow in these patients. So this experiment included 21 sickle cell anemia patients and 13 healthy subjects. And this is a picture of the basic clinical setup. So we used an infrared camera to image the dorsal aspect of their forearm, which is this part of it. And this infrared camera sampled at a rate of 2 Hz, which means 2 pictures per second. Infrared cameras actually measure temperature and not blood flow, but there has been a proven correlation between these two aspects, so we can use an infrared camera to extrapolate our results to blood flow. 20 minutes of baseline, which is just the regular blood flow before we do anything to it, and then we used a cuff to conduct a full blood flow occlusion of the arm for 5 minutes, and so during those 5 minutes we stopped the blood from flowing to the arm, and then we released the cuff and imaged them for another 20 minutes to see how the blood flowed back to the arm. So that is what's known as the reperfusion period. And so here we have the infrared camera, and here we have the cuff. So after we conducted the clinical protocol, we had to process the images before we could get any data from them. So first we had to align the pictures, which basically means if the patient moved at all throughout the process, we had to, so if their arm moved, like jolted slightly, we had to basically place it back on the picture so that each pixel on the picture corresponded to the same region of the body. And after the images were all properly aligned, we isolated a region of interest within the arm, because we were only interested in the blood vessels and the blood flow, not like the whole arm itself. So to do this, we used a thermographic image reconstruction motivated process, which basically isolated the most reactive vessels, and we did this by taking the derivative of the whole the image over time, and then seeing which areas had the highest overall derivative or rate of change of blood flow. So after we found a region of interest, we created a temperature profile graph, and so this is a typical temperature profile right here. And we use these graphs to determine which periods before and after occlusion we wanted to use for our analysis. So this graph is basically the temperature in the pixels in the region of interest averaged altogether at each particular time point. So first we decided we wanted to use the whole 20 minutes of the baseline period, which is shown here before the red line. And then here is the 5 minutes of occlusion where we stop blood flow to their arms. And then for the post-occlusion period, because at the beginning, right when you let the blood flow back to the arms, there's not really much regulation of blood flow. It's just going to the least restricted vessels because there has been blood there for a while and it just wants to get there, I guess. So we were interested in the regulation of blood flow through the previously mentioned mechanisms. So we decided that we would first, we wouldn't use the beginning of the post-occlusion period. So to cut it out, we first found the maximum temperature point reached after occlusion, and then we took the last 90% of frames after the max point. And this is what we called our post-occlusion period. So to analyze the frequency components of oscillations, we used a method called the wavelet transform, which is a relatively new signal processing technique. This technique enables a time frequency resolution of the signal, which means it can tell which frequencies are present at which time points. And this is really helpful because for our signal, it's not really a constant signal, meaning that the frequencies may change throughout time, which is why this method was highly desirable. It compares the mother wavelet to the signal itself at each particular time point and calculates a coefficient, and that coefficient is used to determine how if that frequency point is there at that time point or not. And this is just the equation for the Morlet wavelet here. So after conducting the wavelet transform, we get a wavelet plot. So if this is a typical temperature plot over time, and after the wavelet transform, we see a wavelet power spectrum, and this shows this is frequency in log over time. So the colors represent how prevalent each frequency range is at each time period. The scale is over here, and the from red is really prevalent to not so prevalent in the blue right here. So because it's really hard to tell anything from the graph itself, we used a couple markers to compare between the two groups. The first one we used was average global power, which the global power is the wavelet power averaged over the whole time period you're interested in. And we did this for each individual frequency range. And the second one we used was burst of oscillations, and this is something new that we decided to experiment with because we noticed that over time there would be sudden bursts of oscillation power and then it would die down really quickly too. So we defined a burst to be the top 25% of power within a given spectrum, and we looked at two factors of burst mainly, which is the burst length, which is the beginning of the burst to the end of the burst, and we also looked at burst percentage, which is how much of the total time that we looked at was there a burst. This is a graph of the average global power. So each of the labeled frequency ranges that you see were the ranges mentioned previously and the ones which we averaged the global power over to use for our analysis. Okay, so this graph better explains what a burst is. So I've circled what we roughly define as a burst over here. So you see that before the burst there's relatively low wavelet power, but during the burst there's a sudden peak, and then after the burst it goes back to normal. So this was a feature that we saw in many of the graphs. So we thought this might be something worth looking at. These are the graphs of the average global power for the baseline period and the post-occlusion period between the two groups. So we found that there was a significant difference between the two groups in the NO-dependent endothelial and NO-independent endothelial frequency ranges, but only in the baseline period and not the post-occlusion period. And there was no significant difference between the two groups in the myogenic or sympathetic frequency ranges. You can also note that on average the global power before the occlusion was higher than the power after occlusion. So this graph shows the percent decrease in oscillation power for each frequency range for the two different groups. So we see that for the healthy subjects there was a much higher percent decrease in oscillation power versus the patient group, and this can possibly be attributed to the fact that the blood flow is already restricted or impaired in the patients. Therefore, there's only a certain point to which it can be impaired. It can't just keep going down, right? So yeah. For burst duration, we observed that there was a significant difference in the NO-independent endothelial frequency range after occlusion versus before occlusion, and this was observed in both groups. But we didn't find any differences between the two groups, nor did we find any differences in the other frequency ranges. Burst percentage, we observed a significant difference between the two groups in the NO-dependent frequency range, but we didn't observe any differences before after occlusion or in any of the other frequency ranges. So what do these results mean overall? Well, the fact that we found differences in the NO-independent and NO-dependent endothelial frequency ranges between the two groups in average global power supports previous studies that have shown impaired NO function in sickle cell anemia patients, but furthermore, the fact that we only found these differences in the baseline period and not the post-occlusion period suggests that occlusion may not be a necessary test to study the blood flow of sickle cell anemia patients, which is good because it's a rather painful test to stop blood flow to your arm for a full 5 minutes. In addition, we found that there was a significant difference between the two groups on how much in the percent decrease of oscillation power after occlusion in all of the frequency ranges, and this suggests that blood flow is impaired in the myogenic and sympathetic range as well, at least to some extent, which has not been found previously. Lastly, the differences found with burst of oscillation suggest that it might be a useful factor in studying the blood flow oscillations in sickle cell anemia patients, and this should be further explored. So by further studying these oscillations, we can help lead to better therapies for sickle cell anemia patients and help them better mediate their symptoms and help them lead a better life, basically. And also, the techniques used to study blood flow in sickle cell anemia patients can be used to study blood flow in patients with other diseases that affect the blood flow, such as diabetes, which is a really big disease. So by finding new techniques such as burst of oscillations, we can maybe help those other diseases as well. So in the future, we hope to correlate the factors we found, such as burst of oscillations or average global power, to clinical data, such as maybe heart rate or crisis score, and so maybe these can better predict the specific effects of the disease on each specific patient. And also, we hope to further explore bursts of oscillations by introducing a more robust definition rather than just arbitrarily the top 25% of frequency power, and also maybe look at different factors other than duration and percentage. So I'd like to thank my mentor, Dr. Alexander Gorbach, and my whole lab, which is the infrared and thermometry unit. And I also like to thank the ORWH, FAES, NIH, summer internship program for getting me my internship and allowing me to work there this summer, and also all the teachers who've taught me statistics and biology and computer science and those skills have helped me. So.